Welcome to the BioSalsa podcast, where we host community conversations on the biomedical industry in San Antonio. This $44 billion industry is the largest in San Antonio, the seventh largest city in the U.S. This podcast is brought to you by BioMedSA, a member-driven nonprofit with the mission to accelerate the growth of the healthcare and bioscience sector in San Antonio and the surrounding region. I'm Heather Hansen, president of BioMedSA. We'll explain more about BioMedSA and how you can access the biomedical ecosystem in San Antonio at the end of the podcast.
The American Association for Cancer Research estimates that 20% of all cancers worldwide are caused by infection. Today, we will hear from Dr. Larry Schlesinger, President and CEO at Texas Biomed, who will talk about the connection between infection and chronic disease and the important research being conducted at the Institute.
It's great to be with you, and I'm very interested and happy to speak about what we know in science regarding the robust interactions between infection and a variety of chronic diseases. And it's a topic that I think is very timely for us to discuss today. We'll be talking about different diseases, but I'm going to primarily focus on cancer. And based on your introduction, I would say that we know, and I want to make it clear, not everyone who gets an infection, and 100% of us get an infection in our lifetime, develops cancer. But a subset of us do. And the question is, how do we make that connection? How do we predict who might get cancer following an infection? So to start with, I'd say that when we get an infection, which is a bug, a virus, a bacteria, etc., our body reacts to that foreign invader, and it reacts by activating our immune systems. And when the immune system starts fighting the bug, there's a consequence to that, and it's called inflammation. It's actually the inflammation that gives you a fever, for example, or when you get a cut, builds that redness, and sometimes you get some, pus in the wound. That's inflammation. And what we really want to happen is we want that immune system to kick in, create limited inflammation that resolves the infection. But based on our genetics, our epigenetics, so-called epigenetics, our environment, a number of factors, our age, that inflammation can persist, and it can turn into what we call chronic inflammation. Well, that chronic inflammation is exactly the link that then leads, again, based on our genetics and our environmental factors, to the development of cancer, inflammatory heart disease, coronary heart disease, inflammatory bowel disease, autoimmune disease, and other diseases. So it's critical that we understand that infection can trigger chronic inflammation leading to a variety of those diseases. And Texas Biomed, whose mission is to protect the world from the threats of infectious diseases, by its very nature is also studying those chronic diseases that are inflammatory in nature and can potentially be linked to that infection. So with that, I want to focus on one link that is particularly, I think, important and science really supports, and that is the link between cancer and infectious diseases. And let me start by saying there are several groups at the Institute that are studying infection and cancer, and I'm only going to focus on some research that I'm excited about coming out of my laboratory today. But there are groups studying the interaction between HIV and TB and some factors that can be associated with controlling those infections, and other so-called targets or ways that we can treat infection and perhaps prevent cancer going forward. Now, I'd like to present an analogy. One of the breakthroughs in cancer today is the concept of immunotherapy. And immunotherapy, which many members of the audience may have had themselves for cancer, is the infusion of some molecules, often called antibodies, that are targeting parts of our immune system to activate it. So when a tumor cell or cancer cell starts to proliferate or grow in our lungs or our liver, etc., there is an area around that cancer cell called the tumor microenvironment. That is our immune system trying to wall off and control that cancer. But here's what's important. Cancer cells are smart, and they have developed strategies to turn off the very parts of the immune system that are activated to kill the cancer cell. So the cancer cell, depending on the type of cancer, can win, and that tumor cell can continue to grow while it's shutting off those immune responses. Immunotherapy for cancer precisely tries to wake up or activate those arms of the local immune response to now override the cancer's ability to inhibit it and kill those cancer cells. That's what immunotherapy is. So my laboratory studies an infection called tuberculosis, which is an airborne bacterial infection, and it can land in our lungs, and then it can disseminate in our bodies. It can cause a kind of a chronic infection, primarily a pneumonia, and is really the number one killer of infectious diseases in the world. Now, TB, when it enters our body and it goes into our organs, like cancer cells, our body's immune system attempts to wall it off, just like a cancer. And as a result, we have the equivalent of a tumor microenvironment. In the TB field, it's called a granuloma. And in that granuloma, those immune cells are trying to kill TB, just like it's trying to kill cancer cells. And TB, being very adapted to humans, can shut off those arms of the immune system, just like cancer cells can. There's quite a bit of an analogy there. So what are the arms of the immune system specifically that cancer cells or tuberculosis try to shut off? Well, one of them is a pathway in our immune cells called programmed cell death. This is a natural process in our immune cells that undergo a process of dying, but at the same time can activate the immune system to kill cancer cells. I know it sounds a little bit complicated, but it's the way it works. The ability to induce this programmed cell death, which has a fancy term called apoptosis, the more active apoptosis can occur, the more cancer cells can be killed. But cancer cells, and it turns out that tuberculosis bacteria shut off that programmed cell death. And so it no longer can effectively control the infection or cancer. Well, if there's such a similarity here, we had an idea a few years ago that what if we took antibodies and other small compounds that are in the pipeline of discovery for immunotherapy for cancer to rev up this programmed cell death, to activate it, to kill cancer cells. What if we tried to use these molecules to control tuberculosis? So we literally, contacted other labs. We bought compounds from pharmaceutical companies. We got a whole library of these compounds that can stimulate this programmed cell death pathway. And we asked whether they can control TB. And so it turned out that they're very effective in controlling TB. Now, one important part, and in infectious disease, I should say, we don't say immunotherapy, we say host-directed therapy, or HDT. Pretty much the same thing, only in science we like to come up with different words, so I don't want to confuse your audience, but it's more or less the same thing. So this is HDT for tuberculosis. Now, one of the important elements of HDT for an infection is the fact that normally we treat infection, and you know this, by antibiotics or antivirals, that is, molecules that kill virus cells. But the growing problem we have in the world is that these bugs have become resistant to the antibiotics or resistant to the antivirals. So we're losing the amount of drugs we have to treat these infections. And you may be reading about this in the newspaper. Well, if you use HDT or host-directed therapy where you're boosting the immune system, the idea would be that that no longer is a problem, because whether the bug is resistant or sensitive, that activation of the immune system can kill either. So what we did with tuberculosis is we have in our lab a variety of strains that are either susceptible to normal frontline drugs to treat TB or resistant. And we asked, what's the difference if we activate programmed cell death? That is, we use host-directed therapy. Well, it turned out, as we might have hypothesized, it's equally effective. This is a breakthrough because there's a growing number of multi-drug, extensively drug-resistant strains of TB in the world, and we don't have a lot of good ways to treat them with current antibiotics that you have to take for months and months and months, and they have toxicity. That maybe if we layer on this HCT to the antibiotics, we can shorten therapy. Even beyond that, we might be able to control the inflammation, which means after the infection's control, our body's organs may recover better. Okay, so we have advanced our studies using a variety of novel models in the laboratory that we've discovered using a variety of human cells to test some of these compounds and products that we acquire. This is a nice way to bridge the need to move towards, for example, an animal model. We can reduce the number of animals by reducing the number of drugs that go forward to an animal. And this is really important as we think about alternatives to the use of animals in science. And so we have these so-called bridge models, and we've identified lead compounds that are most effective in controlling TB. And then we've moved some of these studies to these animal models where we have early promising results. So we think we're developing a brand new array of therapies to control drug-susceptible and resistant tuberculosis. We also think this approach will be applicable to a variety of other infections, and we're excited to try them. So that's really, I think, the future direction of my lab, trying to develop novel strategies to treat infection using host-directed therapy, and continue to look at the analogies with, for example, cancer or drugs being developed for other inflammatory diseases. I think in the future, if I was to think big, I would say pharmaceutical companies may no longer just focus on a disease like drugs for diabetes or drugs for cancer or drugs for heart disease, and instead recognizing the commonality with inflammation, we may be looking at drugs that cross disease categories and become more effective broadly. And there's already several examples of that. And I'm excited moving forward that this could be a new era of the way we treat infections that continue to be a global threat and a threat to our families.
Thank you. Can you talk about any of the other important research being conducted at the Institute, either in host-directed therapies or in cancer research?
Well, there is another group at the Institute interested in the co-infections. It's otherwise called as syndromic of HIV and TB. So among those patients with TB, if you're also infected with HIV, the virus that causes AIDS, you are particularly susceptible to a bad outcome with tuberculosis. Another group in our institute has discovered another target, not programmed cell death like we've been talking about, but a molecule with a fancy name, I'll just call it IDO1, which is also found to be highly expressed in those granuloma environments I talked about for tuberculosis, and also is upregulated or increased in its activity in HIV. Well, it turns out that this molecule, IDO1, is an important molecule in suppressing or reducing the immune response in that environment. So kind of like programmed cell death, IDO1 also is a problematic protein because it suppresses our body's ability to fight HIV and TB. And so there are other groups in the Institute that have developed compounds to target IDO1 and inhibit it, reduce it, so that the immune system can be activated. Other work that's going on in my own lab is looking at micronutrients and their ability to control infection as a host-directed therapy. We have a long-standing interest in what are called medicinal metals. These are things that are in our multivitamins that are thought to boost the immune system. And one that many in the audience know about is zinc. And we're actually looking at zinc as a way to control infection. But we're also looking at another metal. When you were in grade school, you learned about the periodic table. This one kind of sits in the middle, called gallium. And gallium is also a medicinal medicine used to treat cancer. And we've been studying it as a therapeutic against tuberculosis. So we have a lot of data to support that packaging gallium in these little packaging lipid molecules can be used as a therapeutic for both HIV and TB. There's another group in the lab that is modifying a known vaccine for tuberculosis called the BCG vaccine, modifying it so it is more effective in treating certain forms of bladder cancer. This tuberculosis vaccine called BCG is a FDA-approved strategy for treating superficial cancers in the bladder. And another investigator at the Institute is studying how to enhance the activity of that vaccine. And then finally, I would say another group is looking at cytokines. These are molecules that float around in the bloodstream and in the tissue that also, when activated, can enhance immune responses to infection, in this case in HIV. And the molecule I'm talking about has a fancy name. I'll call it IL-21. And that is also being used in models in the laboratory and in animal studies looking promising for treatment of HIV and is moving through the pipeline. Remember that a lot of these compounds need to be tested before they ever get to a patient or a clinical trial. So that's where Texas Biomed has its niche in creating new important discoveries that can translate to the clinic, ultimately working with the FDA. So we're in that so-called upstream space with these discoveries. There's another arm of our institute that's very heavily focused on some of these respiratory viruses and trying to identify whether there are strategies that also can boost the immune system against those viruses that are traveling in the community right now and making a lot of us feel under the weather.
Dr. Schlesinger, thank you for your insights. We really appreciate your time today.
My pleasure.
Biomed SA accomplishes its mission by supporting the companies and organizations already here, catalyzing actions for growth that benefit the entire community, and promoting the region and its assets. If you are seeking connections to resources or want to increase your company's visibility in this market, Biomed SA can help. To learn more about Biomed SA, the life sciences in the San Antonio region, or to access additional podcasts, visit biomedsa.org.